Okay, last part of chapter 4. We're going to talk about chloroplast mitochondria and peroxisomes, more specifically chloroplast mitochondria. And we'll actually spend a lot of time on these in class when we talk about their structure and how their structure helps them do the things that they do. So mitochondria and chloroplasts are both found in eukaryotic cells, along with other cellular components. So mitochondria is the site of cellular respiration, not the powerhouse of the cell. It drives me insane when you hear that. Anyway, you can use that if you want, that's fine. So their job is to generate ATP. And what they do is they recycle ADP in AMP into ATP. Right? They do this by taking the energy stored in the bonds of sugar, fats, and amino acids in the presence of oxygen. So another term I gave you in class was oxidative phosphorylation is what they do. It is the site of oxidative phosphorylation. Oxygen, pretty straightforward, that's oxidative. Phosphorylation means to put AP back on to ATP. And chloroplasts literally do exactly the reverse of what mitochondria do. Instead of requiring oxygen, they spit oxygen out the inside. So it's a really cool process, the two of them. So neither one of them are part of the endomembrane system, so they're not part of that system of vacuoles and vesicles and Golgi and ER and smooth ER and the nucleus. They're like literally their own living things. They are these tiny little things living inside of your cells. They've been there for hundreds of millions of years. We evolved with multicellular organisms at the same, actually probably before that. They have their own ribosomes. They have their own DNA. They have their own cytosol or cytoplasm inside. Once again, both organisms have small pieces of DNA. The DNA that they have is, instead of being long and thin like our DNA, it's circular. And it's way, way smaller, has way less genes, and it doesn't change much. So we can look at mitochondrial DNA, and we can compare the mitochondrial DNA of different people from different places around the world. And what we find is we can, like, the whole 23andMe thing that you see on TV, they can look at your mitochondrial DNA and tell where your ancestors came from based on the mitochondrial DNA, not your DNA. So it's kind of a sham in some respects. It's really not 23andMe. It's your circular DNA inside your mitochondria is what they should call it, but that doesn't sound as cool. So mitochondria, almost all mitochondria are found, or molecular are found in almost all eukaryotic cells. I can't think of many eukaryotic cells that don't have them. Pause for 1 sec here. Okay, back at it. So mitochondria have two membranes. They have an internal membrane and they have an external membrane. The reason they have two membranes is because the internal membrane of the mitochondria used to be a cell membrane of the cell that the mitochondria used to be. So let's back this up a little bit. So mitochondria used to live outside of your cells, way, way, way before we were even animals or plants. So at some point along the way, some cell swallowed, let's go back to that last video, phagocytosis took place, and there was a cell that was swallowed. So now the cell had its own membrane, plus the membrane of the cell that swallowed it. The reason that this is important is something along the way happened, and that thing that was swallowed, that bacteria, is really what mitochondria are, that was never digested. Because it wasn't digested, it lived inside of a cell. And just like all cells, the mitochondria where it has its own DNA and does its own thing, they reproduce by splitting into two new mitochondria when they get too big. Just like our cells have to split when they get too big, because cells have to be small. We will talk about some of the pieces and components of the mitochondria as we go. I'll do that more in class. So here's a picture of mitochondria. We have, this is an electron microscope picture. This is a transmission electron microscope. It's looking on the inside, and what you're seeing is you have this outer membrane all the way around, and then there's this inner membrane, all right? And the inner membrane, believe it or not, is all folded like this. And the folds in that inner membrane add surface area. The reason they want surface area is they need area or space to do what they do, and that's to produce ATP. Think of it as like food, some type of food comes in. Doesn't matter what the food source is. The food source is going to contain carbon, hydrogen, oxygen in some variety. That's going to come in with the help of oxygen, and out the other side we're going to get ATP, CO2, and water. which once again we'll talk about more when we get into how this actually works. So we'll draw these. We'll draw this in class. We'll talk about the outer membrane, inner membrane, the cristae, which are the folds, and the matrix, which is this inner space. So this would be like the cytoplasm in here of the original cell that the mitochondria was. Chloroplasts literally work exactly the opposite of mitochondria. Instead of using oxygen, they produce oxygen. Instead of making ATP, they use ATP, right? And they use ATP to produce food, all right? And what they do is they're actually taking the ATP that has been made somewhere else, and they're storing that ATP as a long-term molecule. In many cases, it's sugar. So we'll talk about. There are a couple different types of plastids. I'm really not going to get into amyoplasts too much. These are found in certain types of plants. We will talk about chromoplasts. Chromoplasts are like chloroplasts, but instead of being green, they're either red, orange, yellow. There are those other pigments that you see this time of the year. All right, they also show up in fruits as well. You've probably heard some things like pomegranates, for example, are really good in something called antioxidants. They're healthy for you. Antioxidants get rid of extra oxygen molecules that are inside your body. You think of oxygen as being a good thing, but oxygen in the real world is actually bad in a lot of ways. Oxygen makes things rust. You think of like a piece of metal, it gets wet, there's oxygen around it, it rusts. The same thing happens to you. As you breathe, you oxidize or you rust from the inside out. And by rust, I mean oxygen is stealing electrons. We talked about fat and skinny guys. Oxygen's always constantly stealing electrons from other places in your cells, and it's breaking down your body because it's very electronegative, and it's able to steal those electrons away. So, ironically, some of these chromoplasts are red, so that red coloration you see in a lot of fruits and vegetables, that red color, there's something about that absorbs these extra oxygen molecules that are harmful to you. But we'll kind of get into more of that as we talk about the So it's easier to kind of jump to this picture. So here we have just like in the mitochondria, there are pieces of components that you're going to have to know. So we'll look here. Here obviously is a plant cell. We've got the cell wall here. We know the membrane's just on the inside. We've got a nucleus, water vacuole, ER, rough ER, smooth, mitochondria, Golgi apparatus, vacuoles, vesicles, plasmodesmata. Those are all these pieces. You should be able to identify these. These green structures here are the chloroplast. When you look a little closer, you find that there are these tiny little things inside the chloroplast. Right? These little, they look like little poker chips. Right? They're stacked up. Right? These have a name. These are called thylakoids. The membrane is on this one little piece here, this one poker chip, if you will, is the thylakoid. There are proteins embedded in there. So this thing is made of phospholipids and proteins. The proteins are the pigments. Those are the colors that you see. These pigments happen to be green. The reason you see green is every other color that comes from the sun is absorbed by the green pigment, but green is reflected, hence the fact that it looks green. So that's one membrane. Here's a second membrane. Here's a third membrane. Let's go back here and we talked about this guy. So this was the mitochondria. It has one membrane inside, one membrane on the outside. This was swallowed one time by some organism. What they think here is what happened is this is called serial endosymbiosis. These little guys here, they think, were the actual bacteria that were swallowed once by the first membrane, and then something else came along and swallowed that thing. Right? So the multiple membranes come from being swallowed more than once. Right? Once again, happened a long time ago as cells were developing. You're probably talking somewhere in the neighborhood of 800-900 million years, maybe even 1 billion years ago. This was all happening, and then we've all kind of evolved to this point. You can actually look at mitochondria, and you can say that, hey, mitochondria are older than chloroplasts, and the reason we know this is because mitochondria exist not only in animal cells, but they also exist in plant cells. So from an evolutionary standpoint, there was one type of cell that had a membrane, that had these internal organs and mitochondria. So these guys have both. So at some point, somewhere along the lines, these plant cells differentiated themselves probably by swallowing something that was photosynthetic, in this case, which is these chloroplasts, and then even later on probably developed cell walls. So these are kind of big evolutionary milestones for some of these cells. So here are the parts that you need to know. We'll talk about the fact that the chloroplasts have their own cytosol. They have two membranes. They have these spaces inside. One's called the stroma space, one's called the thylakoid space, or thylakoid membranes as well. We use terms like granum. Granum is a stack of these thylakoids. So here's like, 1, 2, 3 thylakoids. A stack of them would be a granum, or grana, plural. There's also this thing called the stroma. Stroma is the space inside here. But you can see how there's tons of these membranes here. So you have all these tiny little thylakoid membranes. These little stacks are these guys little here. They're all interconnected. They're all interconnected because they share DNA in there. They're able to transport materials from one place to another more effectively. It's kind of similar to like the endoplasmic reticulum in some respects, like that. Once again, has proteins embedded on that, which we'll talk about as we go. So in all of these things here, all these thylakoids are actually giving it more surface area, which allows it to do more of the work that it does. All right, last one we'll talk about is peroxisomes. Peroxisomes are a little different. They are made by the body. They're not made through endosymbiosis. These guys, I'll be honest with you, I don't have a lot of understanding about these. I don't have expertise in these things. But what I will say, though, is that the big thing that they do is they help break down hydrogen peroxide. That's their main goal. But hydrogen peroxide, H2O2, is a byproduct of a lot of the things that happen inside of the body. For example, detoxifying alcohol. If you drink alcohol, which you should not, but I'm sure at some point you will, the reason you're able to get rid of the alcoholic content inside your body, because as your body breaks alcohol down, H2O2 is one of the things that is actually produced. Your peroxisomes help take that alcohol that's in your cells and actually break it down into something that's harmless, which is water and oxygen gas. Some peroxisomes break down fatty acids and other smaller molecules as well, and those pieces then can be transported into mitochondria. But once again, hydrogen peroxide is a toxin that shows up in the body through normal reactions, and this is a way of us buffering or absorbing the hydrogen peroxide that we make as a waste product. We do this with a lot of things. For example, if you're running around the track and you start breathing heavy and you can't get enough oxygen and you start to go into, you end up having too much CO2 in your body, you can become, your body can become acidic. You want to be around pH, 7.2 is about what humans are. If your body gets below 7.2, you can go into a coma. So your body has a way of buffering the acids that are in your body so that doesn't happen. We're able to maintain that homeostasis. The same thing is true with peroxisomes. I guess last piece, once again, they're single bond membranes, so they're more like things that would be produced as part of the endomembrane system, but they're not produced by the endomembrane system. But they look similar to those. They're kind of that in between type of organelle where they're kind of self-made in many ways, but they're not like mitochondria and chloroplasts that have those multiple membranes. And when they get too big, just like other cells, they split in half. Once again, there's a lot more that we can learn about these pieces, but we've done way more with chloroplasts and mitochondria than we have peroxisomes from a study standpoint. So we'll stop here. This will be the fifth video of the little series here. So the last one. Hopefully these help. Hopefully generate some good questions in class.